Okay, listen, you have your second homework assignment, which I forgot to post till a few minutes ago. And then I had to repost it a few shorter minutes ago. But the second homework assignment, you'll be watching what do you call it, a documentary on the history of Earth. And the homework consists of 3 sections that list various events that you have to rearrange in order. Okay, and the way you're going to do that is by watching the video. Okay, so I believe it's 3 parts on the homework. Each part has about 7 events that are given to you, and you have to rearrange them in the correct order. Okay, and you're welcome to take the Word or PDF document and just number them as far as 1, 2, 3, 4, 5, 6, 7 for each block, as far as what event occurred first, second, third, fourth, fifth, sixth. Okay. The videos, it's not a short video, it's over an hour, but it's a good video. So far, most of my students have liked it. Okay, let's go ahead and get going with Chapter 7. Chapter 7 focuses on prokaryotes, or in other words, the domains, bacteria, and archaea. A lot of emphasis is placed on bacteria, but enough will be placed on archaea, and we'll reach a point in the chapter towards the end of today's lecture. We'll do a comparison between those prokaryotic organisms that are classified under domain bacteria versus those that are classified under archaea, not just to justify why they've separated prokaryotic organisms into 2 domains versus prior they were under 1 kingdom, but also to show that archaeans are not only unicellular prokaryotic organisms that aren't exactly like bacteria, but they actually resemble eukaryotic organisms more closely. So prokaryotes in general, they're referred to as the Masters of Adaptations. If you recall, in Chapter 25, we talk about the history of Earth, and we talk about abiotic synthesis leading to the formation of a protocell. Simplest form of a protocell has replicating RNA encased in a phospholipid bilayer, okay, that we call a protocell. And then that protocell advances to where we have other compounds such as DNA and protein, and then eventually we have the evolution of our first living organism, which was believed to be prokaryotic in nature. If you look at the literature, they often believe it's a form of cyanobacteria. But they call them masters of adaptation because they've been around roughly for 3.5 billion out of the 4.6 billion years that the Earth is believed to have existed. And the environment has constantly changed. The Earth has constantly undergone changes. And prokaryotic organisms have proven to be very resilient by continuously adapting to these new environments and undergoing many rounds of speciation. Okay, so they call it masters of adaptation. Now, one of my kids lives up in the Salt Lakes. I didn't get to go look at the Salt Lakes up close, but all I saw was off of a distance, a reflection when the sun set. But the Great Salt Lakes in Utah, they have a salt concentration of up to 32%. This kills living organisms classified under the domain bacteria. They're prokaryotes just like archaeans, but it would kill them. However, for archaeans such as the genus Halobacterium, they not only survive in environments with a high salt concentration, they thrive in these environments. Okay. Now they give the water, such as the salt lakes, this kind of pinkish tint to it. And this is due to a red membrane pigment that's found in the membrane of these prokaryotic organisms, or these archaeans. And that pigment actually functions in protecting these prokaryotic organisms from UV light. But Halobacteria, the genus itself, the most popular species is Linera. Excuse me if I don't pronounce things correctly in the afternoon or noon, but love to live in environments with a very high salt concentration. So other prokaryotes have been found to be tolerant to radiation, strong acidic environments, and extreme temperatures. We'll talk about the ones for extreme temperatures. They're known as thermophiles. If you took 1406 at ACC, you should have covered a thermophile called, what is it called, a TAC, or Thermophilus aquaticus. Okay, that was a type of thermophile, and how researchers identified a type of DNA polymerase that they now call TAC polymerase, and they use it for PCR, or in vitro DNA replication. Now, yet others live in environments that we consider normal. And then prokaryotes are microscopic in nature. Okay, the smallest ones I've come across are roughly 200 nanometers in diameter, or 0.2 microns. Okay, now their ability to adapt to different environments makes prokaryotes the most numerous living organisms on Earth. But they've been divided into 2 domains. Initially, as I've talked about before, they were under 1 kingdom. If you were a prokaryotic living organism, they would place you under the kingdom Monera. But as technology advanced, and especially DNA sequencing and genetic information or genomes go, they realized, okay, just because we have two organisms that are prokaryotic in nature, that doesn't mean they go under the same classification. There are a lot of differences that could not be detected before because of limitations in technology. Well, those limitations now have been surpassed, and so they divided them up in these two domains. So as I said before in chapter 25, they're believed to be the first living organisms, or believed to be prokaryotic in nature. They've gone through natural selection many times over due to constant changes in the environment. Therefore, there's a great degree of diversity for prokaryotic organisms. Most they're unicellular, but some of them live in groups or what we call colonies. Okay. Now, here the textbook says they range in size from 0.5 to 5 micrometers, so I stayed consistent with the text. But technically speaking, if you look them up, I believe it's mycoplasma, I believe, that is as small as 0.2 or 200 nanometers, okay, or 0.2 microns. That's in comparison to eukaryotic cells, with much larger, 10 to 100 micrometers. Now, prokaryotes come in a variety of shapes. Some are sphere-like, which they call cocci. Some are rod-shaped, which they refer to as bacilli. And some look kind of squiggly or spiral-shaped, and they simply call them sporuli. So they come in different shapes. You'll see some of these shapes as we move through the chapter, especially for the bacteria that causes Lyme disease. And then in 1406, you should have gone over the anatomy of a cell in general. Okay, they're often referred as being bacterial in nature, okay, because they're leaning more towards bacteria because you hear about them more. But in general, they'll show you a prokaryotic cell that looks like this. If you recall, they're not only much smaller than eukaryotic cells, okay, but they don't have membranous organelles such as a nucleus, a ER, a smooth ER, and things of that nature. However, they do have their genetic information stored in DNA, and that DNA can tightly pack together, forming a chromosome. But that DNA will be located within a region called the nucleoid. So even though there's not a membranous organelle called the nucleus to store the DNA in, it's still concentrated in a region called the nucleoid. Okay. And when I go over them with my class, I compare and contrast prokaryotes versus eukaryotes. I point out that something prokaryotes and eukaryotes have in common besides DNA, and that genetic information is stored in DNA, is that for both types of cells, you have a plasma membrane. Okay, sometimes I'll just simply say cell membrane, but it's a phospholipid bilayer, entirely made up of phospholipids with many proteins inserted into it, except for prokaryotic cells, because these types of cells tend to exist as unicellular organisms, okay? And even if they're living as part of a colony, okay, they're not multicellular. They're still functioning as individuals, but they live together as a group, so we call it a colony. But since they tend to function as individuals, it's not enough for the outer barrier to simply be made up of that phospholipid bilayer with embedded proteins. There has to be more to protect this living organism. So this is what we're going to get into. We're going to talk about how wrapped around the plasma membrane is a structure called a cell wall. But the molecular makeup of a prokaryotic cell wall is very different than a eukaryotic cell wall. And then when you compare prokaryotes that are classified under the domain bacteria and prokaryotes classified under the domain archaea, you also see there are significant differences in the molecular makeup of their cell walls. However, in general for prokaryotes, cell wall definitely functions in protection. And then for many prokaryotes, not all, but many, they'll have a capsule, and we'll go over that as we continue with these hair-like things, pili, and then we'll go over these structures here called flagellae. So I call it the outer barrier. When I talk about prokaryotic organisms, I don't say plasma membrane or cell membrane, okay? The outer edges or outer boundaries of the cell, I call it the outer barrier because it's made up of 3 layers. Again, plasma membrane being the inner layer. Wrapped around it is a cell wall. Wrapped around that is often a capsule, okay? Very different from what you see from eukaryotic organisms, where it's either just the plasma membrane, or you may find a cell wall around it, but the compounds that make it up are different. So if you look here, an important feature of nearly all prokaryotic cells is a cell wall. Okay, now that cell wall has 3 functions. The 3 you can actually place under the first one. Cell wall helps the prokaryotic cell maintain its shape. The most popular function that you hear about is it protects the cell. And then you could actually put this under cell shape. it prevents the cell from bursting in a hypotonic environment. In other words, in an environment where the solute concentration around the prokaryote is lower than what's in the eukaryote. So that can cause eukaryotes to take on water through osmosis, but that cell wall helps, counter that osmotic pressure or the flow of water so that the prokaryote doesn't balloon up, swell up, and rupture. Now, starting here, bacterial cell walls versus archaeal cell walls, okay? For the archaeans, their cell walls are made up of polysaccharides and proteins, but these polysaccharides and proteins are not interconnected, forming a network that is referred to as a peptidoglycan, but it is a mix of polysaccharides and proteins. However, bacterial cell walls, their cell walls are made up of polysaccharides and proteins, but these polysaccharides and proteins are interconnected, forming a network, okay? A cross-linked network. And I'm going to jump ahead a little bit and then come back. You see these for bacterial cell walls. This is it right here, okay? The little disc-shaped structures, well, 6 sides is letting you know it's probably a monosaccharide. That's the first thing you should be able to recognize, but it's a polysaccharide because they're covalently bonded. So you have 3 polysaccharides coming across, okay, and then interconnecting them are your short stretches of polypeptides interconnecting them. They even give you some of the 3-letter abbreviations for the amino acids, such as alanine, glutamate, lysine, and alanine over here. So this is this is showing you a representation of what we call a peptidoglycan, interconnected polysaccharides and peptides that are networked together. Okay. I put this as a sub-bulletin because you often hear about eukaryotes. Some of them have cell walls. They could be protist to plants, okay? But when you're talking about eukaryotic organisms, okay, the cell walls are made up of either the polysaccharide cellulose or a modified polysaccharide called chitin, which I gave you a picture down here. Because I said I posted an article. If you looked at that current event articles, that's just for y'all to read if you're interested. And they had that study about using chitin, especially to make tools and things. Well, chitin is a modified polysaccharide. Okay, so this is giving you a representation of that. This one is a representation of the polysaccharide used to make, that makes up cellulose that's used to make up plant cell walls. Very different for bacterial and archaeal. However, most often, okay, when you're studying prokaryotes, we tend to focus on the domain bacteria because we come into contact with them very frequently. We even have a symbiotic relationship with them. We do have some with our archaeans, but more frequently we come into contact with prokaryotes that are deemed bacteria. So you hear much more about them. They're often studied more often, and especially in relation to diseases, as we'll talk about as we'll move on through this chapter. So if you look here, there was a Danish physician by the name of Hans Gram. Okay, he came up with a staining technique that allowed him to classify prokaryotes based on the composition of their cell wall. The staining technique he came up with helped reveal that I can have 2 bacteria, and yet the composition of their cell walls will have peptidoglycans, but one of the 2 bacteria may actually have additional compounds contributing to the formation of their cell walls, which actually may provide them with even more protection. So based on Hans Gram's staining technique, okay, which also allowed him to immortalize himself by calling it the Gram stain. I don't know if he's actually the one that chose Gram stain. He was able to use the staining technique, and based on the results, some bacteria would be classified as gram-positive bacteria. If they were classified as gram-positive bacteria, they were described as bacterium that had simple cell walls that contained a large amount of peptidoglycans. However, okay, others that came out a different color from the staining technique procedure, they were deemed gram-negative bacteria. And it was found that those bacteria, based on the gram stain, those that were considered to be gram-negative bacteria, it was found they had a more complex cell wall. Now, the cell wall was still formed by peptidoglycans, but the peptidoglycans only formed part of the cell wall. What scientists eventually discovered is that for gram-negative bacteria, not only do you find peptidoglycans contributing to the formation of the cell wall of these bacteria, but they have an additional component called lipopolysaccharides, or LPS. Okay. Later, it was discovered that whenever you had a gram-negative bacteria that contained LPS contributing along with the peptidoglycan to the formation of the cell wall, they also discovered that LPS was a toxin. So if your immune systems, your phagocytic cells that were capable of eating up foreign bacterium would eat up a gram-negative bacteria, sometimes that LPS that was taken in by that white blood cell could trigger that white blood cell to eventually lead to a very high fever that in the organism's life. So the LPS is a toxin, but it actually is naturally part of forming the cell wall for what we call gram-negative bacteria. Okay. Again, it protects the bacterium, and it's been discovered that when you look at bacterium that are antibiotic resistant, not archaeans, but bacterium that are antibiotic resistant, a larger number of them are gram-negative. Now, does that mean there are no gram-positive that are antibiotic resistant? No, that's not true. MRSA is actually gram-positive, so that's a perfect example. It just means that more of them are gram-negative bacteria that are antibiotic resistant, and it's tied into the more complex cell wall. So whenever you do a gram stain, if you take micro, I believe they do a gram stain at ACC, if I'm correct. Okay, when you do a gram stain, what will happen is if the bacterium is gram-positive, it'll have a violet, purple, some people say blue color. Usually it's violet, okay, is what they'll say, but they may say purple or blue. Okay, if the bacterium is gram-negative, they'll tell you it's pink. Sometimes they'll say it's red. Okay. What I gave you here is one of the staining procedures used. I believe some of my students that took micro that were taking 1407 concurrently, I think they told me they used two different other stains, but the end result was the same colors for positive versus negative. So I gave you a little protocol here, okay, and I'm going to apply it. One protocol I came across for gram staining is they would use a chemical stain or dye called crystal violet, and they would use another chemical stain or dye called fuchsin or fuchsin solution. In between using these two stains, they would use a D-stain or a decolorizer. Okay, so the way it works, all right, so it shows you gram-positive versus gram-negative. The way it works is if it's a gram-positive bacterium, okay, its cell wall is made up of just peptidoglycans. You don't find LPS. So look at this figure here, and they're taking a piece of outer barrier. This mustard colored here represents the plasma membrane, that phospholipid bilayer with embedded proteins. On top of it, this whitish color here, that represents the cell wall. And they're telling you for a gram-positive bacteria, its cell wall is just made up of peptidoglycans. And over here, I'm showing you what they would look like if we could magnify it. Now, what happens is whenever they're performing a gram stain, you initially add, say, crystal violet to the bacterium. You expose the bacterium to crystal violet. That crystal violet, okay, is bound very tightly by the peptidoglycan. Therefore, when you take the bacterium to the decolorization step or destaining step, this compound here called peptidoglycan does not let go of the crystal violet. It doesn't rinse off. So then when the person comes back and adds the fuchsin or the pink colored stain, it doesn't grab onto this because it's already saturated with the violet color. Then when they take it through a second decolorization or second destaining, what ends up happening is it doesn't matter that you expose it to that second color. The gram-positive bacteria has a cell wall only made up of peptidoglycans, and it holds on to that crystal violet no matter what. So you get this color. That's opposed to the gram-negatives. If you look here, you got to look at the order inside out. Again, the inner layer, this mustard color, this represents the plasma membrane, phospholipid bilayer with embedded proteins. Now notice something, the color scheme is different. Out here, the cell wall is this pasty white color. In this figure, you got to look in here. Here's that pasty white color, which represents the peptidoglycan, but sitting on top of that is this wrinkled looking color, mustard color shape, which is this here. The two together, that pasty white color in this outer part, the two together represent the cell wall of a gram-negative bacterium. The inner part of the cell wall that's up against the plasma membrane It's made up of peptidoglycan, but it's much less. It's a thinner layer. Sitting on top of it is the other part of the cell wall for a gram-negative bacterium. It's that lipopolysaccharide, or LPS. Notice, this lipopolysaccharide, known as LPS, it is a phospholipid bilayer like here, like the plasma membrane, except it has many polysaccharides attached to it. Now what happens is when they take a gram-negative bacterium through the procedures of a gram stain, they initially add crystal violet. When they take the bacterium through the decolorization or the destaining step, the chemicals they use for the decolorization or destaining, they're chemicals that you could call lipid soluble. In other words, capable of removing lipids. So what happens is the crystal violet that the bacterium is first exposed to actually grabs onto the LPS. And then when you take it through the decolorization, this is removed. So you lose that violet or purple color. Then when they come in and add the fuchsin, which gives it that red-pink color, now the only thing left after the destaining is a peptidoglycan, and that's what the second stain attaches to. And then when you try to decolor it, it's the peptidoglycan doesn't let go of that second stain. So therefore you end up with gram-negative bacteria that appear pink. Some people will say reddish, depending on how intense the stain color is. But the important part is it's letting a researcher know, okay, that there are differences in the molecular makeup of cell walls just between bacteria, just between prokaryotes that are classified under the domain bacteria. Okay. But like I said, that doesn't mean gram-positives are helpless against antibiotics. Okay, MRSA is the best example, methicillin-resistant Staph aureus. It is definitely antibiotic resistant. Okay, now for many prokaryotes, you will find an additional layer, primarily and often made up of polysaccharides. Okay, often. Okay, and it's wrapped around the cell wall. It's called a capsule. Okay, it's usually made up of polysaccharides. Sometimes it'll be made up of proteins. But the capsule has a couple of functions. One of its functions, and the most popular function, is it helps the prokaryotes stick to substrates or other prokaryotes in a colony. And that's what this figure is showing you here. Here's a cross-section of our bacterium. We've cut through it. You can see the cell wall, and then this fuzzy stuff is the capsule. And they're actually showing you, okay, a prokaryote or a bacterium, that the capsule part of it has helped it attach to the tissues that form the tonsils or the cells of your tonsils here. Okay? But there's an additional function, and it helps further contribute to protecting the prokaryote from our body's immune system as well as dehydration. Okay, but the first function is the most popular for the capsule. Then scientists discovered, okay, once we go through all this and we see they have pretty complex outer barrier for prokaryotes in general, it doesn't matter if they fall in the domain bacteria or archaea, they have a pretty complex outer barrier. Then scientists discovered, and I haven't looked up the history to see who's credited with first discovering this, but it was discovered that some prokaryotes have the ability to form an endospore. It's kind of a hibernation state, if you want to call it, okay? What they discovered is that for some bacterium, such as the bacterium, and they say for prokaryotes in general, but you usually hear it in relation to bacterium. The most popular bacterium, Bacillus anthracis, the bacterium that causes anthrax. Okay, what they found is that some prokaryotes, such as the bacterium Bacillus anthracis, that when the environment is not conducive to just surviving, period, they can go into kind of a how do you say it, a hibernation state or a state of stasis, a state of unchange. What they will do is within the prokaryote, they will start to produce a multi-layered protein coat, which they show you here. At the same time, the prokaryote will make a copy of its DNA, and make a copy of its circular DNA or chromosome that contains its genetic information, and take that copy, okay, and place it within this protein coat. The rest of the bacterial, the rest of the prokaryote, or in this case, B. anthracis, if you want to use it as a specific example, the rest of the body of the prokaryotic organism okay, lysis. It breaks apart, releasing this little structure here called an endospore that has a protein coat around it. Now, what they found is this endospore, okay, it's very durable. It's very resistant to harsh conditions in the environment. And what can happen is that if and when the environment changes to where it's conducive to supporting life, this endospore will regenerate back into a living prokaryote. Okay, so you may have heard with anthrax, there was at one point, the feds were watching the what do they call it, USPS, the United States Postal Service, keeping an eye out for mail that was being delivered to certain people. They wanted to know, were you receiving an envelope with powder inside of it? Well, that's because the powder contained the endospore form of B. anthracis. And they knew that if you inhaled this or got this on your skin, that endospore would regenerate into a living B. anthracis. And B. anthracis produces toxins that can kill you. So we'll go over this guy a little more later in the chapter. If you're interested in these endospores, there's additional information from Cornell. I put a link here, okay? It's got a little bit more information because I have some students that decide they want to go into microbiology, which is a very interesting subject. Every once in a while, you'll find a prokaryote that not only has a plasma membrane cell wall in a capsule, but extending out of the capsule, but coming from the direction of the plasma membrane and extending out of the capsule, it will have these hair-like projections, which are actually protein fibers. They call them fibria, or I call them villi, the easier name to remember. The function of these is to further help attach the prokaryote to a surrounding substrate, or if it's part of a colony, attach to others in the colony that are like it. Okay, so sometimes you'll see this hairy-looking guy. So that's what they're showing you here. Okay, what we're going to transition into, okay, is going to be this region called the nucleoid, as well as we're going to move into the flagellum, or flagella. First of all, motility. Focusing on bacteria, okay, or prokaryotes in general, but it says bacteria, they exhibit taxis. They have the ability to move towards a stimulus, okay? More in general, prokaryotes, both bacterial and archaean in nature, have exhibited what we call chemotaxis. This is the movement towards or away from a chemical stimulus. Okay, are the cells of our own body, okay, within us internally. If you take an endocrinology course or a neurophysiology course, you will see chemotaxis is the key, okay, to cell communication. All right. Now, so you see it also with prokaryotes. Now, most motile bacteria propel themselves by flagella. So they get wishy-washy. They jump back and forth in the chapter, and I've got to fix this to be more consistent. But prokaryotes in nature are found to have not just one flagellum, but often they have more than one flagellum that are scattered about its surface, or you'll find one or more on either end. Okay, this is the thing. When they looked at the molecular makeup of the flagella that found on prokaryotes that are considered bacteria, looked at the molecular makeup of flagella on those prokaryotes that are considered archaea, and then looked at the molecular makeup, okay, of eukaryotic cells that have flagella. What they found is that the molecular makeup was different. Primarily, the protein makeup was different. Therefore, okay, so far, based on what we call proteomics, not just looking at the proteins that contribute to the makeup of flagella on these three different domains, organisms in these three different domains, but also looking at, say, primary structure. When you look at the primary structure of a protein, you're looking at the sequence of amino acids that make up the polypeptide. In other words, what amino acids and in what order, okay, are they arranged? And you start comparing them. You did this for lab 2 with that proteomics or that protein clock. They'll do that. And what they discovered is that there's no direct evolutionary relationship for the flagella located in these three domains, for organisms in these three domains. They're not homologous. So they describe them as analogous. Okay, so again, it says flagella, bacteria, archaea, and eukaryotes are composed of different proteins and likely evolved independently. So to the naked eye, they look the same, but they're not homologous. They're actually analogous structures. They look the same, but when we look at them in detail, they're not the same. Well, we're going to go over this a little bit. So look here, bacterial flagella are composed of three key protein parts or complexes: a motor, a hook, and a filament. Many of the so we're just looking at bacteria right now. Many of the flagellas are proteins, are modified versions of proteins that are believed to have performed other tasks in bacteria. Okay, so in the case of the bacterial flagella, it's believed that the proteins that form the motor, the hook, and the filament of the flagella, the bacterial flagellum, okay, were actually proteins derived from or descended from proteins that were part of an ancestral secretory system. And again, one of the ways they do it is they look at the primary structure and look for homology between proteins that are presently part of the secretory system in a bacterium, and then they compare it to the proteins that make up these three parts of a bacterial flagellum. So they call this exaptation. So here's the working definition for exaptation: where existing structures take on new functions through descent with modification. So it's not just happening at a macro level, it's happening at a micro level. Okay, not just the level of the gene, but the level of the proteins apparently. So this is a bacterial flagellum, and here it is in detail, and here's an actual EM. Okay, here's the plasma membrane or cell membrane. Here's our cell wall. Inserted, okay, cutting through both layers, is the motor here. Then connected here, just above the cell wall, is the hook, and then here's the filament. And basically the way it's described is this motor will turn, thus turning this squiggly thing around. So this thing doesn't move like a tail whipping back and forth, it just spins like a propeller. Okay, that's for a bacterial flagella. To give you an idea of how different it is at the molecular level from a eukaryotic flagellum, using those two as a comparison, use mammals. For mammals, there's only one cell that has a flagellum. It's the sperm in males. Okay. The way it works is you have the plasma membrane forming the outer barrier of the sperm's head, and that same plasma membrane extends out and forms the outer barrier of the flagellum. Okay. Inserted or pushing out that membrane, if it helps think of it easier that way, inserted or pushing the membrane out, creating this tail-like structure, okay, is a large bundle of protein fibers called microtubules that are pushing out. So the flagellum in this case is completely encased in the plasma membrane that also encases the head of the sperm. And then these microtubules cause the entire flagellum to move in separate parts, causing the cell to move. The flagellum doesn't spin around like it does here for bacteria. Okay, so this is what we mean by the structures being analogous. At the molecular level, they're not homologous at all, but they are analogous. Okay, they look the same and they actually carry out the same function, but they're still not homologous because they don't have the same molecular makeup. Bet you more than what you wanted for flagellum, huh? Okay, so the next part here, if you look, internal organization, okay, internal organization in DNA. If you look here, it says prokaryotes are simpler than eukaryotes in internal structures. Now, sorry, I was writing something down. Prokaryotes usually lack complex compartmentalization. Well, I talk about this with my students in 1406, okay? Not only is there an obvious difference in overall size between a prokaryotic cell and a eukaryotic cell, okay? Not only is there a major difference in the outer barrier of a prokaryotic cell versus a eukaryotic cell, but prokaryotic cells don't have membranous organelles. They do have non-membranous organelles, such as a ribosome and a flagellum, but they don't have membranous organelles, such as the nucleus, RER, SER, and Golgi apparatus. Okay. Some prokaryotes do, however, have specialized membranes that perform metabolic functions. And we talked about this going back to the mitochondria, okay, in the previous chapter. What was it, chapter 25? Okay, how we talked about through endosymbiosis, it's believed that a larger prokaryotic host, either through preying on either through predator or through a smaller prokaryote, okay, acting as a parasite, we had a process called endosymbiosis. And when we went over that mitochondria, one thing we talked about is how the inner mitochondrial membrane is infolded. It has a lot of infoldings, and it has a lot of proteins that function as enzymes inserted in it. Well, it comes from the fact that prokaryotes in general, on the inner side of their membrane, it's infolded. Okay, so you usually see infoldings of the plasma membrane, at least on the inner side. And then prokaryotes, I tell my students, when you compare eukaryotic DNA to prokaryotic DNA, it doesn't matter if they're in the domain bacteria or domain archaea. If you compare prokaryotic DNA to eukaryotic DNA is described as linear. It has open ends. Prokaryotic DNA is circular in nature, but often you'll hear it referred to as a circular chromosome, but it is circular in nature. Another difference for eukaryotes, that linear DNA, with the exception of the mitochondria, not including the mitochondria, that linear DNA is stored in the nucleus. With prokaryotes, it's found concentrated in a region called the nucleoid region. Okay. Now, something else you see for prokaryotes that you don't see for eukaryotes is that in addition to the prokaryotes' circular DNA, okay, or chromosome, sometimes they will contain smaller pieces of circular DNA that they call plasmids. Okay. Scientists discovered these a long time ago and started using them for research in labs. They would use them to create vectors. Okay, and sometimes they would still call them plasmids, but it led to a lot of confusions. Eventually, I would say vectors. They would take pieces of this small circular DNA from these plasmids and combine them with small pieces of DNA from other organisms, creating a new circular piece of DNA that I would call a vector. And that's one way they create GMOs, is through this. But the key to plasmids, okay, is one, first of all, they're separate from the prokaryotes' own chromosome, own main piece of DNA, that large circular piece. But they found these plasmids carry genetic information. Some of them are called aplasmids because they carry genetic information in regards to fertility, okay, or reproduction. But the most popular plasmids when I was an undergrad were called R plasmids, resistant plasmids, if you want to call it. These are plasmids because the reason why they were so popular is because they contain genetic information for antibiotic resistance. In addition to that, these small circular pieces of DNA, okay, I'm jumping way ahead. These small circular, oh boy, I'm way, okay, I'll come back to it later. These small circular pieces of DNA can actually be shared between two separate bacteria. So if one bacteria has, or one prokaryote in general, if one prokaryote has this very small circular piece of DNA called a plasmid, say an R plasmid, but another one doesn't, the one that does can actually make a copy of that plasmid and share it with another. We'll go over this, call it conjugation. Okay? So what happened in labs is, like the labs I work in, and sometimes we don't do it with y'all. We do it, I don't know why our non-majors get to do more molecular biology work that y'all should be doing. Okay, they get to do it. But they have a lab, and they use what I call a vector, a circular piece of DNA. And that circular piece of DNA, part of that circular piece of DNA comes from an R plasmid. It was an R plasmid that was isolated, that was found to contain genetic information for resistance to an antibiotic called ampicillin. And what they found is that genetic information in the R plasmid, it coded for an enzyme called beta-lactamase. And what would happen is the bacterium, okay, when it detected that antibiotic was present, or just it produced it all the time. I was under the impression it was just being constantly produced. But what would happen is the bacterium that had that R plasmid containing that gene, that antibiotic resistance gene, would express the enzyme beta-lactamase and secrete it. So that when it was exposed to the antibiotic ampicillin, that enzyme beta-lactamase would literally break the ampicillin molecules in half, thus rendering them inactive, and therefore the prokaryote was immune, was resistant to the antibiotic. Well, researchers realized, you know what, we can take advantage of this in the lab to help us determine, were we successful in creating a GMO? So these became very popular in labs. So ignore this little pill thing, but what they're showing you here in this case is a bacterial chromosome. It is circular even though it doesn't look circular, okay, because it's bundled up. But if you look very closely, you can see here, even here, here, and here, there are 4 small pieces. They didn't point this one out, but there's actually 4 small circular pieces of DNA that are much, much smaller than the bacterial circular chromosome. And these are your plasmids. So they could be carrying more than one. Okay. Now, when it comes to prokaryotic cell division, okay, prokaryotes divide very fast. We call the process binary fission. Okay. Now, when binary fission occurs, it should be reviewed. When binary fission occurs, it's prokaryotic cell division. The bacterium will replicate any circular DNA it has, such as its own chromosome. As it's replicating that circular DNA, it will begin to elongate. It'll separate the replicated DNA from the original DNA, and then the outer barrier will pinch in, and now it's just reproduced on its own. Well, they tend to do this pretty quick. So some key features to prokaryotic reproduction. Well, one, prokaryotes are small. They reproduce by binary fission. And when you put those two together, they're able to reproduce over short periods of time, which they refer to as short generation times. One generation can divide, producing the next generation. That next generation can rapidly divide through binary fission, producing the very next generation. So it's been found prokaryotes can evolve very quickly. Now I need to find this and look it up. I haven't looked it up. But in the text they talk about adaptive evolution in a bacterial colony was supposedly documented over an eight-year period by our research group. Normally when we look at evolution of a species, it's not going to happen in our lifetime. Okay, it's going to be hundreds of years, if not thousands of years, if not millions of years. But these guys have a fast generation time. That means they can produce one generation to the next, to the next, to the next. So supposedly it was detected in 8 years. Okay, the next part. Are there any questions up to now? I know I'm moving fast. You've got an exam, roughly about a week and a half away? Okay, so sorry if I'm just yada-yadding. I usually try to ask more questions. I haven't done a good job of that today. Okay, listen carefully. We talked about systematics. Okay, well now we're getting more specific, molecular systematics. Okay, they're using molecular systematics for prokaryotic phylogeny. In other words, naming and classifying and using it to create a pattern called a phylogenetic tree. Now, until the 20th century, systematics based prokaryotic taxonomy on phenotypic criteria. So that goes back to the fact that if you were a prokaryotic living organism, we dumped you under one kingdom, the kingdom on air. But by applying molecular systematics to the investigation of prokaryotic phylogeny, we've had dramatic results. One of those is that kingdom on air no longer exists. Now we split them up into 2 domains, bacteria and archaea. And they're showing it to you down here. But notice something. If you go off chapter 26, are they showing you sister taxa? Yes or no? If you look at this small phylogenetic tree, are they showing you sister taxa? Yes or no? I mean, yes, I'm finally asking a question. Not rhetorical. Anybody? Yeah. But the sister taxa is not between archaea and bacteria. It's between archaea and eukarya. So if you look here, prokaryotes that were classified as bacteria were found to be more closely related to eukaryotes. Those prokaryotes were reclassified as archaeans. So now when we put the phylogenetic tree together, even though the organisms that are classified under these 2 domains are prokaryotic in nature, with advancements in technology, scientists have discovered actually these guys here are more closely related here. So we put them as sister taxa to the right of the branch point here. Well, what is molecular systematics? Molecular systematics consists of a process called metagenomics. Metagenomics involves techniques such as PCR, automated DNA sequencing, and the use of computer software with algorithms that can help compare the DNA sequences of genomes of various prokaryotic organisms. Okay, so PCR stands for the polymerase chain reaction, and simply put, it's DNA replication. in a test tube. It's in vitro. Except the advantages to PCR is that you choose what DNA you want to replicate, and it's replicated in a very short period of time. So up here, the use of the PCR has allowed for more rapid DNA sequencing of prokaryotic genomes as part of a process called metagenomics. Well, how do we describe metagenomics, or how do we define it? Metagenomics as a whole is the collection and sequencing of DNA from a group of species, a group of different species, that usually come from an environmental sample of microorganisms. Okay? In addition, metagenomics uses computer software to help sort partial DNA sequences and assemble them into genome sequences of individual species that make up that environmental sample. So PCR, I put a little YouTube video on there, so if you want to look at it. PCR is commonly used in research labs, everybody. So if you want to do bench work, definitely watch this video. If you took 1406 here at ACC, normally we would do PCR, but we didn't get to do it in the spring or the summer because we transitioned into online classes. But watch this video, it helps out a lot. It's commonly used. The idea for PCR came from a researcher named Kerry Mullis in the early 80s. Okay, he describes it as he's driving through the hills or mountains of California, and voila, he comes up with an idea of how to replicate DNA rapidly and to choose the DNA sample you want to replicate. And but it still took some time before the technology advanced and the equipment and computer software was developed to where it could be carried out efficiently and without contamination. But by the early 90s, the technique, okay, had been almost just about perfected. A company developed what we call a thermocycler, because in order to perform PCR, you need a piece of equipment that can change temperatures, go through a cycle of 3 different temperatures that promote DNA replication. A huge advancement was the discovery of archaeans called thermophiles, specifically Tachyquaticus, or Thermophilus aquaticus, I'm sorry, which is a thermophile that lives in very, very high temperatures. They identified that it has DNA polymerase like all living organisms, except this DNA polymerase doesn't denature at very high temperatures, temperatures as high as above 90 degrees centigrade. So when they identified that and were able to replicate or produce that enzyme in a test tube, they had everything they needed to perform DNA replication in a tube called PCR. If you do your homework and do your history studies, okay, it's when PCR was developed and performed in a lab efficiently without contamination. Not only did scientists make huge advancements as far as uncovering the DNA sequences and genomes of various living organisms, but forensic science took off, and evidence in criminal cases and paternity testing also made huge advancements. It was because of PCR. I started doing this technique roughly around 94. Okay, but there's something else they're not showing you here. So here it's showing you prokaryotes. When metagenomics is performed, prokaryotes are very diverse and numerous species that inhabit every environment in Earth. So what they discovered is basically if you take a handful of soil, it's probably going to contain at least up to or more 10,000 different prokaryotic species just in that handful of soil that you scoop up. So what they do for metagenomics, collect a sample, and they're showing you different places they could collect an environmental sample. Then what they'll do is they'll extract DNA from any organisms in that sample. They'll take that extracted DNA and they'll perform PCR. Theoretically, with PCR, if you are able to isolate one molecule of DNA from one prokaryotic species, that technique PCR will take that one molecule of DNA and copy it into millions of molecules that are easier to work with and analyze. Then what they'll do is they'll perform DNA sequencing. What they're not showing you here is another advancement that was made, is that when I started doing DNA sequencing about 93, 94, it was a very hammer and chisel type deal. It would take us at least 1 week just to sequence 50 bases of DNA. The thing is, we were working with pieces of DNA that were 5 to 10,000 bases long, and it took us a week to maybe identify 50 bases. It took a long time, and that's if we were lucky. Most of the time, we could only identify 30 to 40. But then I don't know who was credited with this, but somebody developed what we call a DNA automated sequencer. When you look at the DNA automated sequencer, you think you're looking at a computer, a monitor with a keyboard and a hard drive. Well, the lab I worked in, unfortunately, after I was through sequencing, performing DNA sequencing for my research, my supervisor, he had a lot of grants coming in. He was pretty wealthy. That was one of the reasons why I chose his lab. But he bought his own DNA automated sequencer. And when I'd walk into the room and I'd look at it, it has this big clunky monitor that looks like a miniature TV. It has a keyboard. It has a hard drive. But where the hard drive is also, you would remove, one of the techs would remove the front panel. And there was a spot there to insert your samples that were in solution in small PCR tubes. Okay, little tiny tubes that were connected to these small capillary tubes. And my friend would just have to make sure that she combined all the right chemicals in all the right tubes. Don't forget the DNA also. And then she would put them in this little contraption that would hold the tubes connected to these little capillary tubes. She'd slide it into the compartment at the bottom of this computer and hard drive and keyboard, and she'd close the front panel, and then she would hit a key that would tell it to start because it was already programmed like a PCR machine. This automated sequencer did everything else. It would sequence the molecules of DNA that were in the sample. It would analyze the sequences of DNA, and then it would spit out saying, hey, this is what we have. This is what you have. This is how it compares. And it would do it in less than half a day. It would give you everything versus a whole week, you're hoping. On top of that, it would sequence up to 500 bases of DNA in less than half a day, as compared to hoping you got 50 in a week. Another advantage, we didn't have to work with radioactive isotopes. It used fluorescent labels to help you out. So all these advancements, okay, have contributed to a field now that we call metagenomics. And then in addition to that, the college I went to, that I graduated from, the computer science department started working much, much more closely hand in hand with the biology department to bridge the gap between here and here. So you see that more frequently happening. Okay, so molecular systematics is illuminating prokaryotic phylogeny. However, nothing's perfect. There's always a wrench being thrown into everything. And this was the part I was telling you about before. It's a process that sometimes they call conjugation. Sometimes they call it horizontal gene transfer. This is what I was talking about earlier. Sometimes you can have bacteria that are of a similar species in the same genus, okay, and one contains a plasmid that the other one doesn't. It was found that they can actually, okay, develop a connection, a bridge between each other. And the one that has a plasmid that the other one doesn't have, that one will replicate its plasmid and then pass it on through this little opening, through this bridge to the other one. To where now they're both carrying the same identical plasmid with the same genetic information. Okay, this says F plasmid, which has to do with fertility, but if it were an R plasmid, what they would be showing you is this one would go from being antibiotic resistant to both of them being antibiotic resistant. But in terms of metagenomics, it's this horizontal gene transfer or conjugation that is screwing up, creating these phylogenetic trees. So when they start establishing these relationships, they have to be careful, okay? And they have to make sure whoever's writing up the algorithms, creating that computer software, that they're factoring in processes such as this. So I gave you another YouTube link that shows you this process if you're interested, okay? Okay, we're going to get into prokaryotes now, okay? The way it's, I'm sorry, we're going to get into specifics on prokaryotes, sorry. Now for prokaryotes here, we have two domains, domain archaea, domain bacteria. Now the textbook, the 11th edition, the way they go through the domain archaea is they actually don't use these names of what I call clades or groups, okay? They actually instead use the term thermophiles and methanogens, okay? So I'll explain that next week, next class, where these thermophiles and methanogens fall under based on this. However, the text does talk about the 5 clades that make up the domain bacteria. Each clade consists of descendants that are believed to have come from a common ancestor. So we will talk about these in this order: proteobacteria, chlamydias, spirochetes, cyanobacteria, and gram-positive bacteria, okay? But we'll approach it a little different here. Now, before we go into these clades, I want to talk about this. This goes back to why are prokaryotes that are archaeans considered to be more closely related to eukaryotes than to bacteria, but yet we say they're all prokaryotes, bacteria, and archaeans. Well, that's what this table is telling you, a comparison of the three domains. Many prokaryotes that were classified as bacteria, okay, were found to be more closely related to eukaryotes. Those prokaryotes were classified into the archaeans. Listen carefully. The ones you're responsible for going into the test, I placed arrows by. Okay? So if you look at this comparison between the three domains, comparing characteristics or traits, okay, the first one is a nuclear envelope. Well, what is a nuclear envelope to begin with, everybody? That's just something you should have covered in 1406. What is a nuclear envelope? I'm waiting. Yeah. Goes around the nucleus, or more specifically, it's the nucleus of it's the membrane of the nucleus, okay? For why textbooks do this is beyond me. Because instead of saying nuclear envelope, they could have just said what? Nucleus, okay? So I just want to let you know what's going on here, okay? So where it says nuclear envelope, they're talking about the membrane of the nucleus. In other words, when we look at the organisms in the domain bacteria, archaea, and eukarya, do the cells have a nucleus? But instead, they put the name of the membrane. It's absent in bacteria, it's absent in archaeans, but it's present in eukarya because we're referring to a membranous organelle. Then it says membrane enclosed organelles. Well, this should have been put under here with this. So now what they're referring to are the rest of the membranous organelles. Do we have a rough ER? Do we have a smooth ER? Do we have a Golgi apparatus? Are there lysosomes, mitochondria? You can see here they're absent in these two, but they're present here. So far the archaeans seem to resemble those in the domain bacteria more than they do eukarya. But then jump down here to these three. RNA polymerase. When I go over gene expression and I talk about transcription, the enzyme that catalyzes transcription is RNA polymerase. And I briefly tell my students that in bacteria, there's only one type of RNA polymerase. In eukaryotes, there are at least three types, RNA polymerase 1, 2, and 3. We focus on 2, but I tell them just no RNA polymerase. Okay? So that's a major difference between prokaryotes in the domain bacteria and domain eukarya, but look at archaeans. Even though they're prokaryotes, they don't have one kind of RNA polymerase. They also have several kinds like eukaryotes do. So now they have a trait that's similar to this group here. Go down here. Initiator amino acids for protein synthesis. Okay, everybody, when you go over gene expression, you talk about transcription, which is catalyzed by RNA polymerase. Then you go into translation. And you talk about how RNA polymerase synthesizes a molecule of RNA. What's that RNA specifically called, everybody? When RNA polymerase performs transcription, it synthesizes a molecule of RNA, but what do we call that RNA? Because if you don't know, what I'm going to talk about means nothing. And you got a lot of work to do. What type of RNA does RNA polymerase synthesize based on what we talk about when we talk about gene expression? All right, let's try this. I'll give you three choices: tRNA, rRNA, or mRNA. Thank you. mRNA. mRNA. Okay. Okay. Catherine, topoisomerases have to do with DNA replication, okay? But at least you gave me something with nucleic acids. I'm desperate, okay? But listen carefully, okay? So when we talk about gene expression, I focus mainly, I'm a little wishy-washy, okay? And I actually cheat. So I talk about a start codon found in mRNA. That start codon consists of three bases, A, U, G. And those three bases, A, U, G, form a codon that codes for what type of amino acid, everybody? Does anybody remember? when you talk about mRNA, yeah, AG is a start codon, but what amino acid does it code for? It is a start codon, but what does it code for? Yeah, methionine. So this is what I'm getting at here. What I'm asking you is so that you have a better understanding. Now I cheat because if the prokaryotic organism is an archaean, then the initiator amino acid, the very first amino acid added to what eventually will become a polypeptide, is methionine for both eukaryotes and archaeans. However, what I don't tell my students, I let Michael take care of this, is that the very first amino acid, the initiator one, for eukaryotes that are bacteria, okay, is a modified form of methionine called formal methionine. So there is a slight difference, okay? So now all of a sudden we have two traits whereby the archaeans have more in common with those that are eukaryotic in nature as opposed to the beginning. Introns. What are introns, everybody? Anybody remember anything about introns? Okay, not coding DNA, but you're close. Exons are the coding part. Introns then are the opposite. They're the what? Non-coding part. So again, when you talk about gene expression, okay, when RNA polymerase synthesizes mRNA in eukaryotes, as RNA polymerase is making a copy of the gene stored in DNA, making a copy of it in the form of mRNA, scientists found that that eukaryotic mRNA consists of a mix of segments called exons and introns. The exons actually encode for the protein. The introns don't encode for anything, so they have to be what? Spliced out. So eukaryotic mRNA undergoes splicing. Okay? Well, you don't find these introns. It says very rare, but technically speaking, I've never seen anybody prove there are introns in any bacterial DNA, okay, as far as genes go. So it says very rare, but I would put none. We don't tend to talk about introns. In other words, in bacteria, when RNA polymerase is transcribing, making a copy of that genetic information in DNA, but making a copy of it in the form of mRNA, when you read that bacterial mRNA, it reads nonstop how to make the protein. It's continuous. But if you look here for archaeans, you tend to find more frequently these segments, these non-coding segments called introns, okay, more frequently in archaeans. So here you can, it says some, but here you can kind of see it falls in between. And then the last trait, circular chromosome. Well, archaeans have circular DNA just like bacteria. They're all prokaryotes. Eukaryotic DNA is linear. So they say archaeans are more closely related to eukaryotes. Okay, you've got to read through the whole table, but focus on these six. And based on these six, you can see you could have an argument going either way. They're more closely related to bacteria. No, they're more closely related to eukaryotes. They look like they bridge the gap. Okay, so this is just trying to explain a little bit where they're coming up with this. Okay. So if you look, it says domain bacteria. It's divided up into 5 clades. You're going to hear the terms clades and supergroups quite a bit, okay? So bacteria include the vast majority of prokaryotes, of which most people are aware. In other words, they're the ones that we come in contact with the most, so they get all the top billing when it comes to prokaryotic organisms. There are diverse nutritional types of bacteria that are scattered among the major groups. Some are autotrophs, some are heterotrophs, and then they even get into chemotrophs, mixotrophs, and all that. But I just stick with autotrophs and heterotrophs. Now, all prokaryotic organisms that are classified into the domain bacteria, okay, are then classified under one of 5 clades. In other words, we look at all these prokaryotic organisms that we consider bacteria, so we put them under the domain bacteria, but then we start comparing them more closely to look for more homologous features. And based on that, we then divide them up into 5 clades. Well, what is a clade? A group of species that includes a common ancestor and its descendants. So therefore, If you look at chapter 26, wasn't I telling you we had domain, family, class, order, genus, species? And now I'm not telling you any of that. Everything's going through a constant transition. So what I went over in chapter 26, I guess you could call it the old form of naming and classifying organisms or performing taxonomy, where you've got the domain, you've got the family, you've got the class, you've got the order, and so on. But today, scientists are starting to chuck that away, push it aside, and they're saying, hey, we're going to come up with a new form of naming and class. It's still taxonomy, but we're going to come up with new terms. Clade is a very popular one. So the domain bacteria is divided up into 5 clade: proteobacteria, chlamydias, spirochetes, cyanobacteria, and gram-positives. Listen carefully. I'm going to stop right there, unless you want me to keep going. I've got everything spaced. How do you say? I've got everything positioned to where on Tuesday we'll finish up with chapter 27 and get into chapter 28, which is the last chapter that I'll cover before the next test. Your next test is still going to be multiple choice, but you have a much, much larger volume of information to study, and make sure you're listening to these recordings, okay? All right, I'm going to